our keynote session just by going through a few examples of the applications of machine learning to the drug discovery process and how we've been applying this to the H3D projects and pipeline over the last year. So Jess covered quite nicely in the beginning of her presentation some of the types of activities we might commonly employ in various phases of drug discovery pipeline, and how is it so important to try and bring down that excessive cost, both in terms of actual money and time investment. So in this course, we're particularly going to be focusing on tasks that involve correlating structure of chemical compounds with their with some sort of bioactivity or structural property or biochemical property. But in general, machine learning is a really powerful approach because it's a general method for finding a correlation between some input data and its associated output variable. So just for example, we might use this for screening compounds, trying to find the actives and inactives up front so that we don't necessarily have to make and test experimentally all the possible compounds we could make. We could extend this though. We can teach models to try and understand synthetic routes, try and understand which compounds are more accessible to synthesis and those that are potentially difficult. In later studies, we can try and learn properties such as the admin properties, trying to predict how fast a compound might be cleared in the body, or if there are potential toxic flags that we need to screen for or be aware of. And then we can even go more general out of the chemical space itself and think more about how do we design clinical trials, how do we select participants so that we can actually answer our questions, see that a drug works in the most effective way possible. So I'm going to move on to more specifically H3D. So where did this all start? About a year ago, we started our collaboration with Ersilia, so Miguel and Gemma came out and worked with us for 3 months. We designed this project where it was, we looked at the general flow of a screening cascade, an experimental screening cascade for a drug discovery project, which generally follows some sort of activity, measurements. Are we affecting the enzyme or the target we're going for? Then moving on to a little bit of cytotox, or then flags that we need to be aware of, trying to improve solubility, those sorts of chemical properties, and then moving on to the ADME properties. And so Miguel and Gemma worked really hard and developed a really nice model development tool so that we could train in-house models, and we constructed these models and assembled them so that they sort of mirrored a virtual screening cascade mirroring the actual experimental screening cascade. So we could have a set of predictions for all the compounds we might want to make and prioritize those that look most interesting. Now, just to do a whistle stop for some of the projects we've been applying this to, with avoiding as much technical detail as possible for the moment. So using these models, first thing we do is look at the chemical space. So what kind of data has our model been trained on? We're going to talk about visualizing chemical space in the next session, but here all you need to know is that every dot represents a molecule. The red dots are active compounds in our N54 assay. The blue dots are inactive. So this gives us a bit of a distribution of how chemicals are spaced relative to each other. And then when we think about screening new libraries to try and find new chemical hits that might prompt new drug discovery projects, first we need to check, are we in roughly the same chemical space as what our model has been trained on, so that we can rely on the predictions for our new matter that we don't have truth data for yet. So then we pass this through both an in-house model and a model from literature. And what you need to know is that bigger is better, bigger is better. So those that get higher predictions are more likely active, and we can prioritize those to purchase and screen in-house. And that could prompt a new chemical series for further investigation. So then moving on to a chemical series work itself. So here every line represents a compound. In green are the compounds that experimentally we know have better activity values. And on the left-hand side, we see how they were added to our system in date order. So this is now before models were used, project teams understanding the SAR and exploring what is working, what is tolerated on the molecule. And then going back and looking, well, what if we had used a model to try and help us? And we see that in both cases, these are particular chemical series. We would have discovered molecules that look more promising more quickly, which would save us on making all of these extra compounds, and we can get to where we're going much more efficiently. It's important to note that every model is not perfect, so we are going to miss some, and we're going to get some false positives. But on balance, if you can enrich what you're screening, then you're going to be a lot more efficient in your day-to-day. Then we moved on to some more data analysis. So now looking at chemical series that were quite advanced, where we have a few hundred compounds or analogs all within a very similar chemical space, trying to understand how much data do we need to be able to accurately predict a chemical series. So here I've got 3 chemical series, and on the y-axis is model performance. Again, for now, all you need to know is bigger is better. And here we've plotted our 3 chemical series of interest in our Plasmodium library relative to everything else in the library. And we can see 3 general cases. So we've got this blue line, a chemical series, which is really poorly modeled from the beginning, and it's quite difficult to model even with a lot of local training points to that chemical series. We've got a chemical series where it starts quite poor, but as soon as we add just a few compounds to give it examples of what works and what doesn't in the SAR, it improves quite rapidly and stays quite good. And then we've got the dream, I suppose, chemical series that are readily modeled from the get-go, and then in general, more data will always help. Moving on to something a little bit different then. So we've become quite interested in exploring how our bodies process drugs. So most clinical trials, the vast majority of clinical trials have happened in the global north, while the vast majority of genetic diversity is here in Africa. So that there's a bit of a disconnect in terms of how applicable are the doses that we determine for our very well-known drugs to an African population. So here we're going to start looking into how can we correlate pharmacogenetics, so genetic information that affects your gut microbiome and liver enzymes, to see if we can tailor medicines more towards an African context and individual patients. And then something different still, in the bacteria field, they've done studies where they look at drugs, they put them under drug pressure and see how the morphology of the cells might change over time to get an idea of the mechanism of action. Once you know the mechanism of action, you can bring new data to try and understand that perhaps you've got a hit from a phenotypic whole cell screen and you don't really know how it's working. This would maybe give you a direction into the types of pathways you might be looking at in terms of mechanism of action. So we've become interested in, can we extend this to malaria? This is quite challenging because it's not, the cells aren't quite as big as a bacterium cell. Here we're looking at parasites inside red blood cells. And so this is something quite interesting.